Okay, now. We're really excited about the special feature in Molecular Ecology Resources. And to mark the occasion, we're having a special live interview with the wonderful Lua Lopez. So really nice to meet you, Lua. We're really excited about the special feature in the journal. So we're looking forward to getting a bit more behind the scenes understanding of what the special feature is all about and the content of it. So give us a bit of introduction about yourself. That'd be great.
So my name is Lua Lopez. I'm originally from Spain, although right now I'm working in the US. I just started a new job as assistant professor at California State University at San Bernardino, so CSUSB. It's been interesting. Like, definitely I'm loving the job, but there's a bunch of challenges in the current situation.
Absolutely.
But honestly, I can't wait to get things started and like get back to the lab and start like my own research group and start getting students involved in all the research. So far it's been fun. I moved from upstate New York to sunny California, so that's been a change. And I mean, yeah, I got my PhD in 2014. Yeah, 2014. It's been a while. Looking into conservation genomics, and from there I moved to more in like model organisms like Arabidopsis thaliana to try to dig deeper into some evolutionary questions. And like a few years ago, I did a postdoc at Penn State University with Jesse Lasky, which basically is what kind of like trigger all this whole special issue in motion, basically.
Yeah, excellent. So tell me a bit more about how you got to the special issue. So you were obviously inspired by your postdoc, but then obviously now you're taking it a step further.
Yeah, so when I started doing my postdoc, so we were collaborating with people at the anthropology lab. And while I was doing my postdoc, two more postdocs joined the lab. That will be Catherine Turner and Emily Bellis. So kind of like the four of us were thinking that resources are kind of scattered. There aren't those many things available for like non-model organisms or like big mammals or humans. And I'm a bit crazy and decided that if you guys want to join, I would love to do this. I think it will be great to put together a special issue where we can find examples about not only what you can do, but like the new techniques, like bioinformatics and sequencing. They're going like so fast, developing new protocols, new pipelines. So I felt like we needed kind of that space to put everything together. And both Jesse, Emily, and Catherine said, yeah, let's go for it. So the idea was to also not only look into those kind of like the development of the bioinformatics and the wet lab techniques, but also to try to make more visible the resources that we have in natural history collections
That's
to
so cool.
all scientists. Yeah, so like there's a lot of information in there that a lot of people is not aware of it. It's honestly a great resource. It's a treasure. And I think we should be able to utilize it better and hopefully give them the place that they deserve in evolutionary ecology. So that was kind of like the other part of this.
Yeah, for sure. So of all the papers in this feature, can you identify any broad trends amongst them?
Say that again, sorry.
Oh, sorry, no worries. So amongst the papers in the special feature, can you identify any like broad trends amongst them?
Sure. So I think like all of them provide some sort of like advance into the general topic of like using natural history collections to develop evolutionary studies. We find examples from like DNA extraction to the bioinformatics of it, and also like how they can help us in some tricky cases where, for example, like molecular identification of remains, like that.
Yeah.
So, some places where morphology is not enough and we need that extra step. And like all the broader impact that all that can have into just in general the evolutionary history of a taxonomic group, or even just And that's kind of like my soft spot, like the conservation part of it, like the kind of things that we can do to look at how, based on how humans are modifying the environment, how things are changing. And I think that also like some things that can be seen in this special issue, that is that if you look back in time, those results can help us to be better at getting things, like at conserving things in the future. So for me, that's a pretty exciting thing too.
Yeah, for sure. Yeah, it's great that like these resources can be used, and all the amazing past collections can be used in this way to inform future conservation. So that's really neat. So what do you find the most surprising about all this? So running this special feature, what was the most surprising thing you found?
So for me, the most surprising thing when we start thinking about who could be part of these was realizing that these collections can help in so many fields. Like, we're not talking only about evolutionary ecology, which will be kind of like my field, but we're talking about metagenomics, epigenetics, conservation, of course, phylogenetics. So I got really excited seeing some advances in metagenomics and epigenetics, because those are like pretty brand new fields that we are starting to figure out how things are working. So for me, that was also pretty neat. So we just want to show the readers that there is so many things we can do in so many different topics. And then another thing that I think that, like, if you read between the lines, you see that like boundaries are not so strong right now, that you can see how one field gets into another, and how like scientists start doing more general collaborative things than just like, oh, well, I'm a geneticist. Oh, well, I'm an ecologist. So I think you can see that in this issue too, like how using these resources can help you put many pieces of the puzzle together.
Yeah, that's fantastic. So say if you're a researcher, you want to start using natural history collections. How would I, like, so I've never done it before, how would I try to build those relationships with the museums and get started?
So the first thing you need to do is to do a search online. So most museums right now, they have at least a really large part of their collections digitalized. And you can start there, like looking into the different museums. There's also databases for museums where you can just like get the list of all the museums, and you can start going from like the largest ones, because probably they have largest collections, to the smaller ones, but you can be surprised of what kind of small treasures you can find on the small ones sometimes.
Of course.
And just start contacting them, contact their curators. In general, they are like super nice, because they definitely want people to use this, of course, always within some rules, but they want us to use these, because for them it's important, it's their function, it's their service. So that is the first thing to do, just like go online, find the list of those museums, get into their websites, download their digitalized information of the different samples they have, and then get in touch, because they might have more things. And they might tell you, hey, somebody else already did this, so maybe you want to get in touch with this person, because that's another thing. Like, all these data, if we just put it out there, we can do so many things with it. And there is a lot of people working on this, but I think we still don't have like that very tight network of communication. So I think that's the best thing to start doing. And then, of course, if you've never done the wet lab, you're going to need a buddy, because you're going to need help with that. I had no clue about how to do it, and I had a great mentor. She is a postdoc at Penn State, Stephanie, and like, really, she taught me lots of things, and that was the key to success for me to be able to do all those DNA extractions and get them to work. If you have to figure this out on your own, it's kind of like pretty challenging.
So giving your perspective, so what do you think are the big next methodological steps that are needed to really fully expand on your work so far?
So first, I think we need all those collections available online. That's a huge human effort. Like museums need people to get to do that work. But honestly, a large part of what we do is extract information from those vouchers, and those collections, they need to be available so we can understand how to use them and understand what kind of things we can do with them. Then we also need, like, I will say, access to all those samples that have been done already that have not been released. So basically, museums should have, like, if I use the data, if I use a sample, I'm going to give that museum my genetic data, because they should be the ones archiving that. We should have a general archive for all those samples, because museums are like a public service, or at least I see them as a public service. So all that information should be available. So if I want to look at that data from a evolutionary point of view, but someone else wants to look at it from, I don't know, another different point of view, that should be there for everyone. So I think all that information should be public, and that museums should have that space to have all that data stored. Another thing is getting the funding and getting that running. But I think that's the best way for all of us to access that information and to really be good at answering questions, from like being accurate and being able to really go to the teeny tiny detail that sometimes we really want to know.
Yeah. I think it's like leads it to our final question, I guess. So for students, so if you're a student just starting, what's your tips? What's your advice to a student getting involved in genomic research, particularly with natural history museums?
Yeah, so they need to really understand what they are working with. Museum samples are precious. It's something that it's finite. You don't have as much as you want. So you need to be aware of that particular nature of it. You need to be, I think it's like very respectful, might not be the way, but it's like you have something in your hands that it's unique, it's beautiful, and it's precious. So you need to understand that. We cannot just be doing 1,000 tests. Right. You have to test things first. You need to have your protocol in place. You need to know that it works. Of course, sometimes things go wrong, and some samples, they don't work. But first, do your homework, get trained, and then when you can have access to those samples. The DNA of these samples, it's going to be highly fragmented, degraded, and the yield that you're going to obtain, it's going to be low. So you need to be ready for that. And you need to understand really well the protocols to be able to modify them to get as much as you can. Because again, this is something that you're not going to have access to it again. You have access once to a teeny tiny amount of tissue, and that's it. And then when you're doing your analysis, like bioinformatic analysis, you also have to be aware of the nature in terms of genomics of that DNA, because that's going to change many things. This is not going to be a normal pipeline for just like a regular mapping and then a SNP calling. There's going to be lots of little nuances that go together with this kind of data, but that's also the beauty of it, that it's different. So we have to learn how to work with it, and that's what makes it also special. So I think they need to know what they have in their hands. They need to do the homework in terms of getting trained in the wet lab first, and they need to do their research the same way that everybody should do it for any kind of project. But in this case, because again, it's a very precious resource, you have to be extra careful.
Excellent. Well, that's fantastic advice, Laura. And it's been really a pleasure talking to you. And we're really excited about the special feature. I'm looking forward to reading all the papers. And thank you so much for your time.
Thank you so much. It was great speaking to you.
Okay, take care.